Alright guys, welcome back to Underground Science. In this video we're going to be talking about purine synthesis. Okay, so what are the purines we're trying to make? We're trying to make adenine and guanine, alright? Or at least in this case, we'll see later, we're trying to make, let's say if we're trying to make DNA, we're trying to make DAMP, right? Deoxyadenosine monophosphate. And if we're trying to incorporate, or in particular, if we're trying to make RNA, right? Then we would want to get the product AMP, okay? And then same thing with guanine, right? We want deoxyguanosine monophosphate. We're trying to make DNA. And then if we want RNA, we just want guanosine monophosphate. All right, so that's what we're going to try to make in purine synthesis. So let's go and see how that happens. First, it's important to realize that we need a molecule called phosphoribosyl pyrophosphate. Okay, we need that in order to make purines. However, in order to even get PRPP, we need ribose 5-phosphate. And if you guys remember, ribose 5-phosphate is made through the pentose phosphate pathway. All right, so let's go through that at least enough to make ribose 5-phosphate. So remember, first we start with glucose. Glucose can come in the cell and get phosphorylated. Therefore, we'll have glucose 6-phosphate. Let's say it's hexokinase in this case that's phosphorylating glucose with the help of ATP. Right after we have glucose 6-phosphate, remember we can use the help of an enzyme called glucose 6-phosphate dehydrogenase. And remember in this step, we make NADPH. So we go from glucose 6-phosphate to 6-phosphogluconolactone. Then we can use a lactonase enzyme to make 6-phosphogluconate, right? So after we're at 6-phosphogluconate, again, we can use the help of an enzyme called 6-phosphogluconate dehydrogenase. What will that do? That's going to convert 6-phosphogluconate into ribulose 5-phosphate. In that process, we make NADPH, all right? Then from ribulose 5-phosphate, we can use ribulose 5-phosphate isomerase, okay? What this is going to do is this is going to change ribulose 5-phosphate into ribose 5-phosphate. And remember, this is what we need in order to make the PRPP molecule, okay? So let's go see how that happens. All right, so we're at ribose 5-phosphate, right? From here, we can make PRPP, or phosphoribosyl pyrophosphate, via the help of an enzyme called PRPP synthetase, right? We're synthesizing PRPP, therefore it makes sense that the enzyme is called PRPP synthetase. Now, whenever we see synthetase, we can also expect, right, that we're going to be using an ATP molecule, which we do in this reaction. Okay, now we have PRPP, or phosphoribosyl pyrophosphate. From here, we need to make 5-phosphoribosylamine. And we do that with the help of an enzyme called glutamine phosphoribosyl amidotransferase. Okay, glutamine phosphoribosyl amidotransferase. In this reaction, though, when we convert PRPP to 5-phosphoribosylamine, we use a glutamine molecule. All right, so now that we have 5-phosphoribosylamine, from here, we can take 5-phosphoribosylamine, put it through a bunch of reactions, and make IMP, okay? But there's so many reactions after this in order to make IMP that I feel like it's just important to know what molecules we use in the future reactions in order to make IMP. So we need these 6 things in order to make IMP, all right? We need ATP, glycine, glutamine, aspartate, carbon dioxide, and formal tetrahydrofolate. All right, so we need these 6 things and phosphoribosylamine, or 5-phosphoribosylamine, in order to make IMP. Now, this was the important creation, right? IMP is really important because from here, you can take IMP through 2 pathways, right? If we're talking about purine synthesis. One would be, all right, I'll take IMP and make GMP or deoxy-GMP, and I'll take IMP and I'll make AMP or deoxy-AMP, all right? So let's go and see how that happens. So first, let's say we go through this pathway. All right, so we can take IMP and take the help of adenylosuccinate synthetase, or adenylosuccinate synthetase, right? And take IMP and convert it to adenylosuccinate. That makes sense because the enzyme is saying adenylosuccinate synthetase, okay? So that means we're going to be synthesizing adenylosuccinate. All right, cool. So now that we have adenylosuccinate, right, we can make AMP from here. By what? The help of the enzyme adenylosuccinate lyase. Okay, so if we take adenylosuccinate lyase, we can take adenylosuccinate and make AMP. And in that process, we make a fumarate molecule. We let go of a fumarate molecule, which again, remember fumarate, you can use that in the Krebs cycle, for example. And just to be consistent, right, when we're taking IMP, guys, and converting that to adenylosuccinate, we're using an aspartate molecule, okay, along with the help of adenylosuccinate synthetase. All right. So now that we have adenylosuccinate, we can take adenylosuccinate and convert that to AMP via the help of adenylosuccinate lyase, right? And then from AMP, right, we can make ADP, right, adenosine diphosphate, if we just add a phosphate to AMP. All right, and remember when we're going from something that's RNA or non-deoxy to deoxy ADP, right? So this isn't deoxy, it's just ADP, and then we convert that to deoxy ADP, right? Then you have to use the enzyme ribonucleotide reductase, right? That's what we've been seeing in pyrimidine synthesis and purine synthesis. Then from deoxy ADP, you can convert that into deoxy AMP, or deoxyadenosine monophosphate. All right, so we made AMP and deoxy AMP from IMP. All right, sweet. So now let's make GMP and deoxy GMP, okay, from IMP. So if we take inosine monophosphate and we take it in the other pathway, right, we follow it on the other pathway, we can take IMP dehydrogenase and convert IMP, right, inosine monophosphate, to GMP. Okay, sweet. So now we have GMP. And then from GMP, we can just add a phosphate and get GDP, or guanosine diphosphate. And then from GDP, we convert that to deoxy GDP via ribonucleotide reductase. And then from deoxy GDP, we convert that to deoxy GMP by taking out a phosphate, and we get deoxy GMP, right? So we made deoxy GMP, we can make GMP, and then AMP and deoxy AMP, right? Deoxy AMP and AMP, all with IMP. So IMP is an important molecule, right, in purine synthesis. So that's basically purine synthesis for you guys, and I feel like this is important because next we'll talk about certain drugs that inhibit some of these enzymes in this purine synthesis pathway. All right, so I hope to see you guys in my next video. Thanks for watching.